C3 carbon fixation is one of three metabolic pathways for carbon fixation in photosynthesis, along with C4 and CAM. This process converts carbon dioxide and ribulose bisphosphate into 3-phosphoglycerate through the following reaction: CO2 plus a UBP, a 3-phosphoglycerate. This reaction occurs in all plants as the first step of the Calvina-Urobenson cycle. In C4 plants, carbon dioxide is drawn out of malate and into this reaction rather than directly from the air. Plants that survive solely on C3 fixation tend to thrive in areas where sunlight intensity is moderate, temperatures are moderate, carbon dioxide concentrations are around 200 ppm or higher, and groundwater is plentiful. The C3 plants, originating during Mesozoic and Paleozoic eras, predate the C4 plants and still represent approximately 95% of Earth's plant biomass. C3 plants lose 97% of the water taken up through their roots to transpiration. Examples include rice and barley. C3 plants cannot grow in hot areas because RUBIS co-incorporates more oxygen into RUBP as temperatures increase. This leads to photospiration, which leads to a net loss of carbon and nitrogen from the plant and can, therefore, limit growth. In dry areas, C3 plants shut their stomata to reduce water loss, but this stops CO2 from entering the leaves and, therefore, reduces the concentration of CO2 in the leaves. This lowers the CO2-O2 ratio and, therefore, also increases photospiration. C4 and CAM plants have adaptations that allow them to survive in hot and dry areas, and they can, therefore, outcompete C3 plants in these areas. The isotopic signature of C3 plants shows higher degree of 13C depletion than the C4 plants. See also, C2 photosyntheses. References